will be going to talk about ATP. As we all know that ATP is the energy of the cell. Cell needs energy for various purposes because the cell has to do metabolism. So it has to build up things and break down stuff. It has to replicate. It has to maintain itself. So repair work is also done in the cell and many other processes. So all the biochemical processes that happens in the cell. In each and every process the energy is required, and the energy providing molecule of the body, ATP, is also known as the energy providing molecule. So energy molecule of the body is ATP. And it is also known that the ATP can be consumed in very high amounts in everyday work. So in a whole day, around its own body weight of ATP is consumed. Okay, body weight of ATP is consumed. So ATP is required by the cell for these various purposes, and the ATP production, the center for production, the powerhouse of the cell where ATP is produced, is called as mitochondria. So inside the cell there is a specialized chamber or specialized organelle, if you go into the scientific terms, and it is called as mitochondria, where production of ATP takes place. So it looks after every energy need of the cell because it is the main powerhouse of the cell. We can also say it as a powerhouse of the cell. So why ATP? The question comes, why ATP and why not any other molecule? So the answer this question, because the ATP molecule, it is a nucleoside which is without the phosphate plus the 3 phosphate groups, the triphosphate. That's why we call this adenosine triphosphate. So the nucleoside plus the triphosphate, the third phosphate bond, which is, we can see here that 3 phosphate groups are linked with the adenosine nucleoside. So the third phosphate bond, this one, is very high energy bond. And when this bond breaks, it releases a lot of energy. And this is the property why ATP is used and not any other molecule is used, because it can be readily easily converted into ADP, which is adenosine with the 2 phosphates. So it is a non-charged ATP, we can say. It is adenosine diphosphate. Adenosine diphosphate. And through the biochemical energy, through the energy of electrons in the mitochondria, we can build up easily the ATP molecule. So ATP can be built up very easily inside the mitochondria. Okay, so I'll draw the structure of mitochondria. What does the mitochondria look like? And this whole process takes place when we use or consume sugars, lipids and proteins in the body. So when we digest the sugars, when we digest the proteins, and when we digest the fats, okay, we use the energy from the digestion of them to build up ATP inside mitochondria. So it is somewhat like an elongated organelle, okay, and it has an outer membrane, we can see here, and it also has an inner compartment where the actual production of ATP takes place. So this is an inner compartment, and we can see here that in mitochondria this is the outer membrane. This one is the inner membrane. And these folds which we see in the mitochondria, they are called as cristae. And the function of this fold is to increase the surface area. So they increases the surface area. So why this increase in surface area is useful? The increase in surface area is useful because it provides a increased amount of effective area. So more area for a larger production of ATP. ATP production by a single mitochondria. Okay, and each and every cell is not limited to a single mitochondria. The cells which require more ATP, like liver cells, the hepatic cells, the liver cells can have around 4,000 mitochondria per cell. Per cell. Okay, so it depends on the functioning of the cell, how the cell functions, how much energy it requires. According to that, the number of mitochondria are regulated inside the cell, and the number of mitochondria can be easily regulated. It can be up regulated or down regulated as per the energy requirements of the cell. And inside the inner membrane we have the matrix. We can see here that this is the matrix. So matrix can be like called as a cytoplasm of the mitochondria, because it contains all the proteins, all the enzymes which are required for the production of ATP. And between the outer membrane and the inner membrane we have this region, and this whole region is called as the intermembrane region. So this is the intermembrane region, and this region helps in providing the gradient which is required for the production of ATP. So later in the video we will understand what this gradient is and how this intermembrane region plays a role in providing a gradient for the production of ATP. So now we'll understand how production of ATP takes place inside the cell. So because we are aerobic organisms, which means that we require oxygen to live, to function. Our cells require oxygen. So have you ever wondered why this oxygen is needed? Later in this video we will discuss why this oxygen is needed by us, by all the living creatures, and why it is very useful for life to sustain. Aerobic creatures use oxygen, the cells use oxygen to function, and oxygen is required as a electron acceptor inside the production of ATP. So we will discuss it in more detail in this video, why this oxygen is needed. Production of ATP in aerobic cells is through the process called as oxidative phosphorylation. So phosphorylation is the process, enzymatic process, which means to add or remove phosphates. So addition of phosphates to a compound is called as phosphorylation. So when we add phosphate to ADP, which is the diphosphate, we make it a triphosphate. So 3 phosphates are added and we make up ATP. And in the process we use oxygen, that's why it is called as an oxidative phosphorylation. And this process is very effective. Oxidative phosphorylation alone can produce around 30 to 38 ATP by consuming a single molecule of glucose, right? So 6 carbon sugar can yield around 30 to 38 ATPs. So how this carbon, the C6 carbon or the glucose, undergoes the oxidative phosphorylation, we can see it in a brief. First, what happens is that the glucose molecule, which is the 6 carbon sugar, the C6, undergoes glycolysis. So it undergoes glycolysis, and the glycolysis takes place in the cytoplasm of the cell. So it happens in the cytoplasm of the cell, and glyco means glucose and lysis means the breakdown. So the breakdown of glucose takes place inside the cytoplasm of the cell, and after glycolysis, two C3 molecules or 3 carbon molecules are made, and this C3 is pyruvate. Then this pyruvate moves to the mitochondria. Inside the mitochondria, which is the matrix, so it goes inside the matrix of the mitochondria, and inside the matrix the pyruvate get converted into a C2, which is acetyl CoA. So one more carbon atom is lost, acetyl CoA is made, and now what happens is that it enters into a cycle. So the C2 enters a cycle which is called as a Krebs cycle, and the C2 gets added to a C4 and results in a C6 molecule, which is a citrate molecule. So it is a citrate molecule, and this cycle is called as a Krebs cycle. Krebs cycle, or also a citric acid cycle. So this Krebs cycle or the citric acid cycle, why it is called as a cycle? Because no reactant is used or it is removed from the cycle, it is regenerated in a cyclic form. So that's why it is called as a citric acid cycle. The citric acid cycle, the citrate in the citric acid cycle, it breaks down and forms some other products and again returns back to the C4 state and captures in one more C2 acetyl CoA, becomes again the citric acid molecule, and again the cycle runs. And after this, the products of the Krebs cycle, we can see that some products will be formed. I will discuss the products in the video. These products will be used in the process of oxidative phosphorylation. So oxidative phosphorylation takes place now, which results in the formation of ATP as we have discussed. Okay, so the products now leads to the oxidative phosphorylation, and it happens through ETC, which is the electron transport chain. So ETC is called as the electron transport chain. And due to this, the oxidative phosphorylation takes place and ATP molecules are formed inside the cell. I will not go into the detail of glycolysis, Krebs cycle. I will make future videos for that where we'll be going into detail into each and every step for glycolysis and Krebs cycle. I will just make an overview picture how a glucose molecule results in the formation of ATP. And when it undergoes glycolysis, so it undergoes glycolysis inside the cell, it results in the formation. So when a glucose molecule it undergoes glycolysis, it results in the formation of some ATP in it. So some of the ATP is provided. So ATP production does take place, but this is not oxidative phosphorylation. This ATP formation under the glycolysis, it is not due to oxygen molecules, but it is through another process. So this ATP forms through the substrate level phosphorylation process, and the substrate level phosphorylation is not very effective process. Only 2 to 4 ATP molecules are produced, and we can see that in each and every enzyme which happens to take place in the glycolysis cycle, only one ATP molecule is produced by the substrate level phosphorylation, whereas in the oxidative level, the oxidative phosphorylation, multiple ATPs are produced in a single step. Okay, so glycolysis do provide with some ATP, so some energy is provided by the glycolysis cycle in the cell cytoplasm. And when the pyruvate, which is the product, so this pyruvate moves to from the cytoplasm, moves to the matrix of the mitochondria, it again gets rid of another carbon molecule and becomes C2, which is the acetyl CoA. And the enzyme used in the process is called as pyruvate dehydrogenase, and these enzymes are very special. The dehydrogenase enzymes are very special because they are the redox enzymes. Okay, so they are the redox enzymes. They carry out redox reactions, which is reduction and oxidation reactions. So dehydrogenase is a special kind of redox enzyme which produces the chemical which is NADH. So why this NADH is important? See, the chemical produced NADH is nicotinamide adenine dinucleotide, and so this NADH is important because it acts as an electron carrier. Because while breaking down glucose in the whole process of glycolysis and in the whole process of Krebs cycle, electrons are released and many electron carriers are produced. What these electron carriers do is that they carry out the electrons, they take them inside the mitochondria, and inside the mitochondria they release their electrons to the ETC, the electron transport chain, which further transfers its electrons to the final electron acceptor, because the carrier, the donor, so it can also be called as an electron donor, and the donor has to donate its electrons to the electron acceptor in the final stage, and the terminal or the last electron acceptor inside the cycle, which results in the production of ATP, is called as oxygen. So the oxygen is the final electron acceptor inside the whole cycle, and it is a key step where NADH is produced. C3 carbon is converted into C2 by the enzyme pyruvate dehydrogenase, which carries out a redox reaction. And whenever we encounter the enzyme dehydrogenase, we have to keep in mind that whenever we encounter the word dehydrogenase, it has to work as a reduction or an oxidation reaction. It is not that dehydrogenases only produces NADH, the process can also be reversed. So NAD molecules plus the electrons results in the production of NADH plus a single proton, and this process can also be reversed. So this process can also be reversed, and another product that is formed, and another electron donor that is formed inside the Krebs cycle, is called as FADH2, which is a flavin adenine dinucleotide, and it is different. It is also an electron donor, so it is also a donor, and it is different from NADH is that NADH is more high energy. So NADH is high energy as compared to the FADH2, which is somewhat lower energy than the NADH. So a single NADH, because NADH can donate 2 electrons, whereas a single FADH, I'm not talking about the FADH2, can donate only a single electron. That's why I'm saying that the NADH is high energy, whereas the FADH2 is low energy. Okay, so next we move into the Krebs cycle after the conversion from pyruvate to acetyl CoA. So now we have the acetyl CoA with us, acetyl CoA inside the matrix of the mitochondria, and now it incorporates itself into the Krebs cycle. As I said, the C4 mixes with the C2 because this is a 2 carbon compound, makes a C6 in a cycle which is a citrate or a citric acid. That's why it's called as a citric acid cycle also, and the cycle churns on, and the products of the cycle are NADH and FADH2 and more ATP. So this ATP production is also through the substrate level phosphorylation process. So it's not oxidative phosphorylation, it is the substrate level phosphorylation. And we can see that the products that are found, these are the electron donors. Now they have to donate their electrons into the electron acceptors, and the electron acceptors come in the form of ETC, or the electron transport chain. So this is the electron transport chain. It is called as electron transport because the electron is transported in a continuous fashion from one enzyme to another or one protein to another. And what happens is that the transfer of electrons, let's say from NADH to the first factor and the second factor, because 4 factors are there in the electron transport chain, and in the 5th process the ATP is produced through the process of oxidative phosphorylation. So these are the 5 sequences through which the electron jumps from NADH to the final, which is the 5th part where the ATP is produced. And the NADH is the electron donor, so this is the electron donor, and these all are the electron acceptors. But the electronegativity of each, the first complex, the second complex, the third and fourth, it increases. So electronegativity increases. That's why the electron gets transferred easily from the first to the second and so on. So electronegativity of the complexes increases, and in the final step, which is the 4th step, before the production of ATP, the electron gets transferred to oxygen. So here we know the use of oxygen, which is the most electronegative. So it is the most electronegative, the oxygen is most electronegative. So that's why we use it in the whole oxidative phosphorylation process, because it acts as a terminal acceptor. So oxygen acts as a terminal acceptor because nothing is more electronegative in the ETC than the oxygen atom. So oxygen now accepts the final electron, and the oxygen atom combining with the electron makes H2O. Okay, so finally when electron is accepted, it makes H2O. So 2 electrons are accepted by the oxygen molecule and water is produced, which is the final product of the ETC, the final product. So in the electron transport chain, as I have talked about, that there are 4 complexes. We can see here that the 4 complexes are here, and the electron jumps or hoops from one complex to the other. So it moves on because the complexes are increasing in electronegativity. And what happens next is that in the process, when the electrons are jumping, the complexes, they act as pumps. So they pump out the protons. So they pump out variable number of protons inside the intermembrane space of the mitochondria. As I was saying, that intermembrane space is very useful in the production of ATP. So here is the mitochondria, and here is the inner membrane with all its folds, the cristae. Okay, so the complexes of the electron transport chain are present on the mitochondria itself. So we can see here is the complex 1, the complex 2, the complex 3 and the complex 4, okay, and so on. And all these complexes, what they do is that they pump on protons into the intermembrane space, so increased concentration of protons. So what this does is that it creates a proton gradient. What does this term means? That the concentration of proton is greater in the intermembrane region. The intermembrane region has higher concentration of protons as compared to the matrix of the mitochondria, which has the lower concentration of protons. So it causes a gradient, it makes a difference in the proton concentration. Next, what happens is that these protons are useful to run the complex fifth of the ETC. So complex fifth of the ETC is different from every other complex, because after complex 4 the electron jumps into oxygen and forms water. So what is the use of this complex fifth? The complex fifth is the key element of the ETC, which is called as an ATP synthase. So this is the molecule which causes the oxidative the phosphorylation, the addition of phosphate to ADP is done by this enzyme, which is the ATP synthase, which means that it synthesizes ATP. It synthesizes ATP. So it grabs the ADP, which is the diphosphate, and the inorganic phosphorus, and forms ATP. And to form ATP, protons are required. So protons are required to form ATP. So now you get my point. Glucose molecule undergoes glycolysis and then it goes through the Krebs cycle just to form NADH and FADH2. So everything in the cell is done, the glycolysis and the Krebs cycle, they are done to just form the proton donors. Once these proton donors are produced, they go to the ETC, and in the ETC they help in transporting the electron, and the energy of the electron is used by the complexes, to pump out protons into the intermembrane region of the mitochondria. And once these protons form a gradient, means that the concentration of these protons gets higher as compared to the matrix of the mitochondria, these protons return back into the matrix. So the protons, they return back into the matrix, and during this process the ATP synthesis takes place by the complex, the fifth complex. So now I'll give you the overview how this all works. So we have the complex 1 here. So these are all the transmembrane proteins, so they are embedded in the membrane or the crystal of the mitochondria. Okay, this is the complex 2, this one is the third complex, here we see the fourth one. I will not get into the details in this video, the details are left out for future videos, so please check them out on my channel, which will give you a detailed overview of the glycolysis, the Krebs cycle and the electron transport chain. So now comes, which is the complex 5, which is the ATP synthase. It is different from other complexes is that it does not require electrons to function. So the electron after complex 4 gets transferred to oxygen, as I have already told you. ATP synthase doesn't require electrons, it only requires protons. So it has a structure of a motor. So if it has one transmembrane region which passes through the membrane of the mitochondria, the inner membrane of the mitochondria, this is called as an F naught, and down is a cylindrical part which is called as an F1. So the F0 and an F1, they combine to form an ATP synthase, which is required for the synthesis of ATP. And what happens is that it works like a motor. Okay, so this part, it twists, it turns. The F1 part turns. What happens is that when the protons that were present, the gradient of the protons that was present in the intermembrane region, the energy of the proton now turns the motor, turns the F1. When the protons passes through F0 to the F1, it turns the F1, turns the motor, and the motor action converts the ADP plus the inorganic phosphate into ATP. So this motor is very uniquely designed. In the first step, it requires 3 protons to make a single ATP molecule. Okay, in the first step, what happens is the first proton is used to attach the ADP and the inorganic phosphate to the F1, the ATP synthase. The second proton is required for the production or the combination of ADP and inorganic phosphate into ATP, and the third proton is required for the release of ATP. And when ATP is produced inside the mitochondria, it is exported out from the mitochondria into the cytoplasm, and the concentration of ATP remains higher in the cytoplasm, and the concentration of ADP and inorganic phosphate, which are consumed when the ATP is consumed by the cell, they get converted into these byproducts, and the concentration of the ADP and the inorganic phosphate is higher inside the matrix of the mitochondria. So the matrix of the mitochondria in itself is a different cytoplasm of the organelle, where the concentration of ATP is lower, whereas the concentration of inorganic phosphate is higher. So you can see here is the complex fifth. So the protons that are pumped by these complexes, we can see the protons, they rush back inside the ATP synthase. So now you get the point how ATP synthesis and why it is called as oxidative phosphorylation, because oxygen is used in the process as a terminal electron acceptor, and then ATP is produced. NADH can hold on to 2 electrons. NADH can hold on to 2 electrons. So effectively it can churn out around 3 ATP molecules per NADH, whereas the FADH, because it holds on to FADH2 holds on to 2 electrons, but FADH can hold on to a single electron. Effectively it gives out 2 ATP molecules. So for each and every NADH that is produced inside the cell, 3 ATP molecules are produced, and for every FADH2 that is produced, 2 ATP is produced. But theoretically, this is a theoretical assumption, but in practical situations, because there are losses inside the cell and not everything is perfect, so instead around 2.5 ATPs are produced, and by FADH2 around 1.5 ATP is produced. That's why we say that in oxidative phosphorylation around 30 to 38, because we take some window of efficiency in it. So we say that around 30 to 38 ATP molecules are produced by a single glucose molecule. So single glucose, which is the 6 carbon molecule, gets converted into a 30 to 38 ATP, and that's how the energy currency is produced inside the powerhouse of the cell, which is the mitochondria of the cell. So it was very brief overview. I have not got into any of these complexes, how these work, and any of the scientific nitty-gritties of the whole process. I will discuss the process again in future videos. This was just a basic overview of the whole process. and thanks for watching my video. Please like and subscribe to my channel